So, good evening all. I welcome all of you to our online master course Plant Sciences 2024. So, we are dealing with plant breeding course. So, I am your mentor, Roopa. I have completed my undergraduation from College of Agriculture, BC from Mandya, which is under the University of Agricultural Science, Bangalore. So, I have completed my master's in genetics and plant breeding from Indian Agricultural Research Institute, New Delhi. So, I have honored with 8 gold medals during my UG. So I have secured All India rank of 6 during my JRF examination, and also I have cleared ASRB net, and I have got SRF rank of 28 in my SRF examination. So today's topic of discussion is domestication and conservation of the germplasm. So let's get started. So yesterday we have seen that about the domestication. So what do you mean by domestication? Anybody?
Cultivation of plant species or a wild crop under human management.
Yeah, good. So it is nothing but bringing the wild species under the human management. So in BDC, we can see as an example for recent domesticated species that they have given some of the examples. I'll just mention those examples. I have mentioned those examples here. You can read those examples. Those are important. So under the domestication, you can see Jatropha, which is used as a biofuel crop, which is one of the important example they might ask it. Okay. And coming to Oryza alta, this is the very much important example. So Oryza alta, it is an allotetraploid which is having a CCDD genome. You have to remember this. Okay. It is also having characters of increased vigor, genome buffering capacity, and greater environmental adaptability. Okay. Oryza alta, remember. And also one of the domesticated species is a beauty palm in Brazil. Okay, these are some of the example. You can go through it. Okay. So next, coming to selection. What do you mean by selection? Anybody?
Okay. Differential reproduction rate.
Yeah, good. It is a differential rate of reproduction of the different genotypes. So under the selection, we can see the types of selection. So what are the different types of selections? Anybody? So we have natural selection and artificial selection. What is natural selection and artificial selection? What is a major force which act in natural selection? Okay.
Environment.
Yeah, it is a environmental forces. Like it could be a climatic changes, or it can be a soil factors, or it can be pest and diseases. So it is a natural force which act to select the genotypes. Okay. So in artificial selection, we can see it is through the human force. Okay. Human force will be involved in the selection. So here, what happens to the variability? How the variability is influenced in this natural population under natural selection? So variability is retained. Okay. So in artificial selection, variability decreases. Why? Because as we go on breeding and breeding, so we will be selecting more of the homogeneous genotypes, right? So we will reduce the variability. And so where do you see this natural selection? It occurs in the natural population itself. Okay. So this is under the crop cultivation practice. We will go for this artificial selection. These are the two kinds of selection you can see. Okay. So natural selection acts mainly by the two mechanisms. What are those? Anybody know? Two mechanisms of natural selection. So one is environmental selection. Another one is a competition. What is environmental selection? So all those genotypes which are unable to cope up with the environmental change. So environment will be continuously changing. So those genotypes which are unable to cope up with those changes, they will be eliminated gradually. That is environmental selection. So what about competition? So competition is nothing but it occurs in the croplands. So in the similar genotypes, when a field is surrounded by the similar genotypes, those plants which can able to absorb more of the nutrients or which can grow vigorously, they will outcompete the others. Okay, that is the competition among the genotypes. This is the two ways through which the natural selection will act. So you can see selection is a differential rate of reproduction as we have seen. So you can read this difference between the natural and artificial selection. So next part is the types of selection. So what are the types of selections? Anybody have read this? Types of selection?
Directional, stabilizing, and disruptive.
Yeah. Directional selection. Stabilizing selection. And disruptive selection. So what do you mean by directional selection?
Ma'am, that favors extreme phenotypes.
Yeah. So selection that favors the extreme phenotypes. So your graph will be like this. Only the either you will select either the tall or dwarf you will select. That is selecting either of the extreme. That is your directional selection. Okay. What kind of gene action you can see in this? Dominance or recessive? What nature?
Dominance or epistasis.
Yeah, it is majorly dominance and the epistasis we can see in this. So next, and I'll give you these two traits. Fertility. So what do you think? What kind of selection might act in these traits which are associated with viability and fertility? Anybody can guess?
Directional.
Yeah, it is always a directional selection for the traits associated with the viability and the fertility. Okay, because always the nature selects the more fertile one, more viable one, which can cope up with its changes constantly. Okay, it is always a directional selection. So next, coming to stabilizing selection. So what is stabilizing selection?
Ma'am, it favors intermediate phenotypes.
Yeah, you can get the graph like this. So your intermediate phenotypes will be more than either of the extremes. Okay. So does it favor dominance or epistasis?
Ma'am, dominance in both direction or additive gene action.
Yeah, not dominance in both the direction. It is additive gene action. It favors the additive gene action. Okay. So the next kind of selection is a disruptive selection. So what is disruptive selection?
Ma'am, it is for those, it's a kind of directional selection, but for those phenotype that are dispersed.
Yeah, it is a selection of the extreme phenotypes. So you will have both tall and the dwarf genotypes. Okay, selecting both the extremes. So it is also favored by the dominance and the epistasis.
Ma'am, then what is the difference between directional and disruptive selection?
You will select any one of them, but here you will select both of them. That is the difference. Okay.
Okay, ma'am.
Yeah. And here you will maintain the polymorphism. Here there is a loss of polymorphism. You will continuously going to select the tall ones only. So you will lose the dwarf ones. Here you will maintain the polymorphism in the population. It is just like a simulation of the natural population. So you will have the both types. That is the difference. So this is the graph you can see for the three kinds of selections. This is for the directional selection. So the mean of your population will shift in the direction of your selection. Okay. So this will be intermediate selection, and this is either of the extreme phenotypes will be selected. So, and you have to remember this, viability and fertility related traits will be directional selection. Okay. So under the stabilizing selection, it is favored by intermediate phenotype, acted against the extreme phenotypes. It will always act against the extremes. Only the intermediates will be favored. Okay, it will disfavor the dominance, accumulate the alleles for the additive gene action. This is important. Next, disruptive selection. So both extremes you will be selecting, diversity or polymorphism will be maintained, and it also favors the dominance and the epistasis. So the last point is disruptive selection lead to the establishment of the supergene. What do you mean by supergene? Anybody know? Supergene. Have you heard of it? Anybody?
Yes, ma'am, but I didn't understand it.
Supergene is nothing but it is a set of closely related genes which together lead to a establishment of a optimal phenotype. Okay. So this in detail, I have to read few more literatures to get a clarity about this, why disruptive selection is leading to the formation of supergene. I'll explain you in the next class, okay? So you keep this point. They might ask you directly that disruptive selection, or they might ask as a set of closely related genes that together lead to the development of a specific optimal phenotype. They can give you in this kind of question, blank. So it is a supergene. They are the closely related genes which leads to the development of the specific optimal phenotype. Example is a male and the female form. In the next class, I'll give you a detailed idea about this, okay? So next, coming to domestication syndrome traits. So as we have seen, we are domesticating the crops from many years, okay? So while domesticating, we can see that our crops have changed with certain traits. Okay, those traits we call it as a domestication syndrome traits. It is like a set of, or what do you mean by syndrome? Anybody? What do you mean by syndrome? It is a
Accumulation of symptoms.
yeah, group of symptoms. So here also we are meaning by syndrome, we mean that it is a set of traits that associated with this domestication phase. Okay. So if we look into this, there is an elimination or the reduction of the shattering of the pods, okay, or the spikes. In particularly in case of brassica, wheat, barley, we can see this. And also we have reduced the dormancy in the species wherever dormancy is associated, and we have reduced the toxins, BOA, erucic acid. So we have modified the plant type according to our need, that is their altered tillering nature or their leaf characteristic or their height. Okay, it has come here. Reduced plant height in case of cereals. So green revolution is also associated with this reduced plant height. And also in case of millets to reduce the lodging. So increased plant height. Where do we need increased plant height? It is in case of jute, sugarcane, and forage crops. So change in photoperiod sensitivity to make them able to cultivate in their out of their original area. Okay, where they are adopted, we can cultivate outside of that area through this change in photoperiod sensitivity, making them as a photoperiod insensitive. So increase in the size of the grains associated with increase in the yield, fruit as well as the economic yield. And also we have focused with shortening its life cycle and shift in the sex form. Wherever we need more of the male flowers or more of the female flowers, we have changed it. And asexual reproduction, polyploidy, species origin. Okay, these are all the traits which are associated with the domestication. So these set of traits together we call it as a domestication syndrome traits. Okay. So next we'll see the pattern of evolution. So what are the 3 patterns of evolution? Does anybody know about this?
Mutation,
Yeah, mutation,
hybridization,
hybridization,
and polyploidy.
polyploid. Okay. So in case of mutation, it is also called as a Mendelian variations. Okay. So we can see 2 types of mutations in this, macromutation and micromutation. What is macromutation and micromutation? So macromutation is the one which gives us a large evident variation in the crop species. Okay. So it is believed that our today's maize is originated from the
Tripsacum.
grassy pod corn. Yeah, it is also considered as a tripsac. Okay, through a single macromutation, our today maize has been originated. There is a speculation that saying there is a single macromutation which has changed this grass into a today's maize. Okay, micromutation. What do you mean by micromutation?
Change in single base pair.
Which have small, less effect.
Less drastic effect. It won't be having any drastic effect. Okay, how they will be retained in the population? Micromutations. Through the natural selection. Through natural selection, these micromutations persist in the nature. So the second pattern of evolution is the hybridization. That is an interspecific hybridization. So one of the example for this, sorry, interspecific hybridization is, can anybody say what is the example for this? So there is a example that there was a primitive maize was there. When it is hybridized with a tripsacum, it is believed that our modern maize has originated. Okay. So many other examples we can quote for this interspecific hybridization in case of sugarcane, in case of cotton that we have seen in the yesterday class. All those examples are the patterns of evolution under this hybridization phase. Okay. So polyploidy. What do you mean by polyploidy?
More than one genome
Or
set.
that one gene affecting
Yeah.
something.
Complete genome will be in a 2 or more sets. Okay. Autopolyploidy, allopolyploidy. So what are the examples? Under the autopolyploidy, we can have autotriploids, tetraploids, hexaploids. So quickly quote the examples for this. Who knows?
Apple,
Yeah, apple, banana. Next,
watermelon,
watermelon and sugar beet. For autotetraploids,
potato,
you have rye, potato, alfalfa, and grapes. So you have to remember these examples. They are important. Okay. So autohexaploid,
Wheat,
this is an aloe. Wheat is an aloe. Okay. sweet potato is an example. So under the allopolyploids, what are the examples? There comes two types, that is a natural one and artificial.
Wheat,
Wheat.
triticale,
Triticale is a natural or artificial?
artificial,
Yeah, it is not a natural. You have cotton, brassica species, tobacco, oats. So you have many examples. I have listed them. You can go through. Under the artificial hybrids, we'll see in there only. So you have three kinds of evolution pattern: Mendelian variations, interspecific hybridization, and polyploidy. So as I told you, Mendelian variation is the macro and micro mutation which is accumulated over a period of time. So under the interspecific hybridization, it is crossing between the two different species. So what do you mean by introgressive hybridization? As the name says, it is the introgression of some of the genes which we need from one species to other species. That is a introgressive hybridization. So autotriploids we have listed, autotetraploids and autohexaploids we have listed. Allopolyploids, you know, it is a different species. From the different species, two or more complete set of genome will be there. So for natural allopolyploids, cotton, wheat, brassica, tobacco, oat will be there as example, and artificial allopolyploids. So yesterday we have seen this example, Raphanobrassica, which is produced by Karpachenko. Okay, it is a cross between the Raphanus sativus and Brassica oleracea. What is Raphanus sativus? It is radish
and
cabbage. Unfortunately, he didn't get a good result for this. And one more artificial allopolyploidy was produced by Clausen and Goodspeed. This is also very important. Yesterday also I have told you. So he has crossed between the Nicotiana tabacum and Nicotiana glutinosa to obtain the Nicotiana diglota. It is a hexaploid. Okay. So another examples for the artificial allopolyploid is a triticale. Triticale is produced by Rimpo. Okay, it is a cross between the Triticum aestivum and the sickle cereal. What is sickle cereal?
ray.
Yeah, it is a ray. So through colchicine doubling, he has produced this. In wheat also, they have attempted this polyploidization by McFadden and Sears. It is a cross between the Triticum turgidum and Triticum tosti, which has resulted in a hexaploid wheat, which is very much similar to this Triticum aestivum. These are all the kind of examples you have to remember. Under cotton, so we have our American upland cotton, which is a Gossypium hirsutum. It is actually a cross between the Gossypium herbaceum and the Gossypium raymondi. Okay. So next we have germplasm, about the germplasm. What do you mean by germplasm?
Collection of diverse allele.
Collection of
Diverse allele.
diverse alleles. Anybody can define it more precisely?
Sum total of all the alleles.
Okay. Where? From where those sum total of all the alleles?
In a species, in a cultivated species, or in its wild forms, all the genes and its allele.
Yeah, good, very good, very good.
Germplasm.
Both in cultivated and its wild species. Okay, set of all the hereditary material, whatever it is there, those hereditary material which is present in the cultivated as well as this wild species, we call it as a germplasm. Okay. So germplasm can be of two types. It can be of a cultivated germplasm or a wild germplasm. So as the name says, which is present in the cultivated species, it is a cultivated, which is present in the hereditary material which is present in the wild species, it is a wild germplasm. Or it can be of another two types, that is a indigenous or wild. Sorry, exotic. Okay, indigenous is from our origin itself. So when we don't have any germplasm collection, we will import it from somewhere else. That will be the exotic collection. Okay. So this germplasm is also called as a genetic resource. Who has given this? So this term genetic resource was coined by, anybody have read about this? It is Sir Otto Frankel, who has said that germplasm is nothing but genetic resource. So when you say germplasm, what it is composed of? So what are all the materials you can classify under the germplasm? Or what are all the materials that are included in the germplasm?
Land races, breeding lines, wild relatives, obsolete varieties, also cultivation varieties.
Yeah, very good. Land races, absolute. Wild forms and weedy relatives. Modern cultivars. Breeding lines. So what are land races?
Primitive cultivars.
Yeah, those are the primitive cultivars. What are they? How they are important for us as a germplasm?
For resistance.
So
High level of genetic diversity.
yeah, they are characterized by the high level of genetic diversity, and they are the storehouse of many valuable genes. Okay. They have evolved under the natural conditions, either naturally or artificially. So while their evolution, there is no systematic breeding effort is involved. So they have occurred naturally and they have persisted naturally. So they're well adapted to the adverse condition. They are well adapted to the adverse condition so that they'll give you a stable yield, but it will be low. Since there is no improvement for the yield, they'll give you a stable yield even under the adverse conditions, but that yield will be low. They are the storehouse of many of the valuable genes. This is important. So what are obsolete varieties?
Varieties of recent past. Which are replaced by
Yeah, very good. These are the varieties of the recent past. That is, once they were very famous, but now they are of no use. So examples are NP series of wheat, K65, K68, I guess. So those are the examples. So what are wild forms and wild relatives, VD relatives?
Ma'am, species from which cultivated species are directly originated, and while wild relatives are the common ancestry with that cultivated species.
So I have the three terms here. So anybody can differentiate what are they? Someone was telling.
Ma'am, wild forms are the from which cultivated species are directly originated.
Yeah, very good. From which directly today's cultivated species originated.
Ma'am, and wild relatives are the species that have common ancestry with that cultivated species.
Yeah, all other related species for this present day cultivated species. So what are VD forms?
They currently compete with crop species when they are cultivated.
Okay, VD forms are the bridging species between the wild form and the present day cultivated species. Okay, present day cultivated form. They acted as a bridging species.
Hello, ma'am.
Huh?
Can repeat VD forms again?
VD forms are nothing but they are the bridging species between the wild forms and the today cultivated forms. They are more like their relatives. Okay. So directly gene transfer has not occurred from this wild form to today's cultivated form. In between, we can see this VD relatives. That's how the croplands have originated. So next one is a modern cultivars. What are they?
Currently cultivated high-yielding varieties.
Yeah, they are the currently cultivated high yielding. What is the nature of those materials? They are highly
high
homogeneous.
yield potential.
Yeah, they have a variability for the yield or
Narrow
high
genetic
yielding.
base.
They have the narrow genetic base. Okay, and they are more of homogeneous materials. Breeding lines. What are breeding lines? And we have another one is a. What are breeding lines?
Pre-release plants.
Okay, while breeding, you will develop set of homogeneous or homozygous lines by repeated selfing or something. What that is the breeding lines. It could be a mutant line which you use in the further breeding program, or it can be a homozygous lines which you use in your breeding program. All those lines which you use for breeding that is available with the breeder, that is a breeding lines. They are also considered as a under the germplasm. So special genetic stocks. What are the materials which come under special genetic stocks?
Transgenic lines.
Yeah, transgenic lines. Transgenic lines and the another
Lines
example,
Chromosomal.
chromosomal
Chromosomal.
aberrations.
lines carrying the chromosomal aberrations. These are the special genetic stocks. All these materials are considered as a under the germplasm. So you have your absolute varieties. This you guys can read. Special genetic stocks, VD forms, and breeding lines. So next we have the gene pool concept. So who has given this gene pool concept?
Harlan and DeWitt.
Very good. Harlan and DeWitt. So what do you mean by gene pool?
Sum of the all allele and their genes.
Yeah, all the alleles of all the genes. Present in what? This is important. Present
In an individual.
no.
Which are hybridized with each other.
Yeah, in all the individuals which can hybridize with each other. That is important. Okay, that is a gene pool. So we can see three kinds of gene pools. What are those?
Primary,
Primary, secondary,
Secondary and the tertiary gene pool. What are the natures of primary gene pool?
Released, right?
All the cultivated crop plants and their wild relatives or their progenitors, not the wild relatives, their progenitors will come under the primary gene pool. So how is the hybridization? Whether they will yield us fertile hybrids or what?
Fertile.
Yeah, hybridization within the members of the gene pool 1 is easy, and we will get the fertile hybrids when we go for this hybridization. Okay, fertile hybrids we will get. So if you quote the examples for this, Oryza sativa, Oryza nivara, and Oryza rufipega. So these are all the species which comes under the primary gene pool. What is secondary gene pool? So those species which when crossed with the members of the GP1 will result in a partially fertile hybrids. They are considered under the secondary gene pool. Why do you think there is a partial fertility? What might be the reason for this? Anybody know? Due to the ploidy difference, it could be due to the ploidy difference or chromosomal abnormalities, which lead to the partial sterility or partial fertility. So coming to, so examples for this is a Oryza barthi, Oryza glumipatula. I have quoted all the examples for this, you can read. So tertiary gene pool. What is tertiary gene pool?
When GP3 crossed with GP1, infertile lines.
Sterile.
Sterile.
Yeah, we will get all the sterile hybrids. So crossing is possible between them. Here, crossing is possible, but we will get the partially fertile hybrids. So here, crossing is possible, but we will get the sterile hybrids, and crossing is possible through some special techniques. Okay. What are those special techniques which we can use? Here also same thing we can see, ploidy difference or chromosomal abnormalities will lead to the sterility. So with certain special techniques, we can make able to cross GP3 with the GP1. What are those techniques? That is either embryo rescue we can go for, or bridging species we can use while crossing, or ploidy manipulation, so that there won't be any ploidy difference between them. So these are the special techniques we will use to make them possible to cross with each other. So there is one more gene pool. Anybody is aware of it? Anybody knows?
Quaternary gene pool.
Yeah. Quaternary gene pool. What is it? What materials are included in this?
Transgenes, transgenic breeding.
Yeah. Transgenic lines are involved in this. It can be called as a gene ocean. This gene pool is called as a gene ocean. Why do you think so? It is called as a gene ocean. Because it is like an ocean. You can bring the gene from wherever you can. Wherever you want, you can bring your gene through this transgenic technology. You don't have any limit for this, right? That is why it is also considered as a gene ocean. So how does it was possible? Through the rDNA technology, it was made possible. What is rDNA technology?
Recombinant DNA.
Recombinant. Can you elaborate what we will do in this? Oh, just a minute.
Hello, students. I think our internet connection is lost. I believe she'll be joining soon. Just wait for 1 or 2 seconds.
Is my screen share has stopped, sir?
Yeah, it has stopped.
Okay, I'll share again. Sorry for the disturbance. Okay, is it visible?
Yes, ma'am.
Okay, sorry for the disturbance. Okay, we were seeing quaternary gene pool. So it is also called as a gene ocean. We were seeing gene pool concept. As I have seen, it was given by Harlan and DeWitt. So gene pool is all the genes and their alleles which can hybridize with each other. So we have seen primary gene pool, secondary gene pool, and the examples are important. You can remember these examples: Oryza bartri, Ruma patella, Oryza longistamina, meridionalis, and glabrema. They comes under the secondary gene pool. So tertiary gene pool is the crossing will be difficult, but you can achieve this crossing through the ploidy manipulation, bridging crosses, and embryo culture. So, but the crossing between the GP3 and GP2 is comparatively easy as compared with the GP1. So all the wild species of the Oryza other than AA genome will comes under this. So species in the related genera of the tribe Oryzae will also come under the tertiary gene pool. So quaternary gene pool, it is GP4. It is also called as gene ocean. So through the rDNA technology, it was made possible. So this is how your gene pool will looks like. So GP1, GP2, and GP3, and all the examples for them. So next we have the concept of germplasm conservation. So before that, why do we need germplasm conservation? Anybody?
In
What
order to retain variability.
Okay. Anybody else?
Saving diversity.
In order to counteract something, we are going for conservation. What is that? It is
Genetic erosion.
Genetic diversity.
very good, very good, very good. It is genetic erosion. So to stop this genetic erosion only, we are going for this conservation practice. What is genetic erosion? Come on, quickly.
Loss of variability.
Okay, you are all aware of it. Good. So it is a loss of variability due to the replacement of the heterogeneous materials from your homozygous or homogeneous pure line or whatever improved materials. That is a genetic erosion. So as we can see the causes for this, it is a genetically variable land races will be replaced by your uniform pure line. And also you will be breaking this crop weed complexes. As you told, as I have told you, so VD complex act as a bridging species between your wild and the today's cultivated form. That is how the gene transfer has happened. So in the management practices, what you will do, you will eliminate all these weeds. So there you are breaking your crop weed complex, which was helping you to transfer some of the genes. But now they're not there. That is how you are unknowingly making this genetic erosion, and also from your grazing or farming or accidental introduction of the VD species, which will lead to the competition and elimination of your crop species or other VD forms or wild forms. So germplasm conservation. For this, we have to do this germplasm conservation. How do you do this germplasm conservation? There are two methods of conserving the germplasm. What are they?
In situ and ex situ.
Yeah, in situ and ex situ conservation. What is in situ conservation?
Within natural habitat.
Yeah, within its natural habitat will conserve. What is ex situ?
Conservation away from natural habitat.
Yeah, very good. It is conservation away from its natural habitat. So what are the examples for in situ conservation? Where do we see this in situ conservation?
Natural parks.
Yeah, natural parks. Very good. Next. Anything?
Gene sanctuary.
Gene sanctuaries.
Biosphere reserve.
Biosphere is very good. So how many natural parks are there in India? Any idea about this? How many natural parks are there? One not 6 natural parks are there. How many gene sanctuaries? So we'll talk about gene sanctuaries later. Biosphere reserves, how many are there? Any idea about the number? 18 are there in India. Okay. So gene sanctuaries. So there are two famous gene sanctuaries. What are they?
Citrus gene sanctuary in Meghalaya.
Okay. Where is the place? You remember any place name?
Garo Hills of Meghalaya.
Yeah, good. It is Garo Hills of Meghalaya for citrus conservation. And one more is there. What was that? Northeastern region. It is for conservation of oryza and banana you can say Musa, banana, and also for the citrus and mango saccharin species. So for this, we have another gene sanctuary, that is a northeastern region. So what is ex situ conservation and what are the kinds by which we will do ex situ conservation? We have 5 modes for this.
Orthodox seeds.
That comes under the seed gene bank.
Gene bank. Seed
Seed
bank.
bank. Yeah. So I think we have this. Can you mute yourself? Shesh 3. Okay, field gene bank. Seed
Cell
gene
and
bank.
organ gene bank.
Yeah, very good. Cell
Shoot tip gene bank.
organ gene. Shoot tip.
Cryo bank.
Shoot tip. Okay. One more.
Meristem gene.
Meristem and shoot tip are same.
Cryo bank.
DNA.
Okay.
Cryopreservation.
It is DNA gene bank. So we can quickly see this under the seed gene bank. So virtually you can say all your conservations are majorly through this seed form. Okay. It is a seed gene bank. All your conservations are majorly concentrated as a seed. So under the seed, we have 2 types. So what are those? Who has classified this seed?
Roberts.
Very good. Roberts has classified the seeds into 2 groups. What are they?
Orthodox and recalcitrant.
What are orthodox and what are recalcitrant?
ऑर्थोडॉक्स का मैम 5 डिग्री टेम्परेचर 5 डिग्री मॉइस्चर में अप टू -196 टेम्परेचर.
Come again.
Orthodox should have 14%.
Up to मैम 5 डिग्री मॉइस्चर में 5%
Without losing their viability.
But it is not 5 degree, it is 5%.
5%.
Yeah, you have to be precise. 5% moisture.
Moisture content.
It can be stored below the 5% moisture. That means it will tolerate the desiccation. And also it can be stored under the sub-zero temperature, that is below 0 degrees Celsius. That is, it is a freezing tolerant. So those species that are desiccation as well as the freezing tolerant are comes under the orthodox seeds. So what are recalcitrant?
Up to 30% moisture content. Storage 12 to 30%.
Up to 12 to 30%. They won't survive if their moisture content goes below 12 to 30%. So, and they are not tolerant if they are stored under 0 degrees Celsius. So they have to be stored at around 10 degrees Celsius. Okay. They are not desiccation tolerant nor freezing tolerant. So what are the examples?
Rubber, coconut.
Yeah. First,
Mango.
for the orthodox seeds.
All field crops.
All your field crops, food grains, legumes, those comes under orthodox seeds. And what are recalcitrant? And examples for recalcitrant?
Horticultural crops, mango.
Citrus.
Mango, citrus,
Coffee.
coffee, very good. Any other?
Cocoa.
Cocoa,
Jackfruit.
jackfruit. Very good. You guys remember the examples also. So we have in situ conservation and ex situ conservation. So we have seen all the natural parks and biosphere reserves, gene sanctuaries, and we are seeing all this gene banks. So coming to your orthodox and the recalcitrant seeds. So orthodox seeds can be taken as a long-lived seeds. So they are the short-lived ones. So they are not desiccation tolerant or freezing tolerant. They are desiccation tolerant as well as the freezing tolerant. They are not tolerant to desiccation as well as the freezing. Okay, examples you can read and remember. So apart from these two kinds of the seeds, we have one more seed, that is a intermediate seed. We'll see that. So intermediate seeds are like, they can tolerate the drying. It is like a orthodox seed, but they cannot tolerate the freezing. It is like a recalcitrant. That's why they are called as an intermediate. They behave both as a orthodox seed as well as a recalcitrant seed. So they could be stored under 45 to 65% RH, and they can be stored for 5 years when they are stored about minus 20 degree Celsius. What are the examples? Neem, umbrella tree, key apple, and papaya will comes under the intermediate type of the seed. This is another class of seed. Okay. So next we have the types of collections. So we have the germplasm collection. So what are the types of germplasm collections we have with us? What are the 3 types? So it is base collection,
Active collection, working collection.
active collection, and
Ma'am, working collection.
yeah, very good, working collection. So quickly
Core
define
collection.
what is base. A core collection, we'll see it later. So what is base collection?
Ma'am, for long-term conservation.
Principal.
Yeah, very good. It is also known as a principal conservation. So it is for a long-term storage. What is a year? Anybody remember? How many years?
Ma'am,
we
up
can
to
store it?
50 years or 50 to 100.
Yeah, up to 50 years or above 50 years we can store. So what else we have in this? So up to what temperature they are stored with what moisture content?
minus 20 degrees Celsius with 5% moisture.
Yeah,
content.
minus 18 to 20 degrees Celsius they are stored, and up to 5% moisture they can be dried. Okay, so for this base collection, we will have one backup collection. Why so? Because all our germplasm is concentrated in this base collection. If by any of the disaster, if this base collection gets destroyed, all of our germplasm will get lost. Okay, for that we will have a backup collection. Where it will be there? Anybody remember the place? Svalbard Global Seed Vault. It is also called as a Doomsday Vault. You remember this. This is a backup collection for our base collection. So what is active collection? These are the medium storage collections, which can be stored at 0 degrees Celsius and with a moisture of 8%. So what is the number of year it can be stored? 10 to 15 years it can be stored. So it is like it is actively used in the breeding programs like their multiplication, evaluation, their distribution. For all those things, we will use the active collections. So what is working collection?
Ma'am, for short term, 3 to 5 years.
Yeah, good. It is for short term, 3 to 5 years. With the temperature?
Ma'am, 5 to 10 degrees Celsius.
Yeah, 5 to 10 degree with a moisture of about 8 to 12%. So 8 to 10% moisture we will store. So it is also known as a breeder's collection. Why?
It is frequently used in.
Yeah, it is frequently
Used in crop improvement.
Crop improvement. This is the collection which will be with the breeder. He will keep this materials and he will use continuously in his crop improvement programs. That is why it is known as a breeder's collections. These are the 3 types of germplasm collections we have. So you can read in this. So next coming to your field gene bank. So what is field gene bank? It is nothing but, as the name says, in the field, you will have the crops as your germplasm. It can be used in case of fruit trees and the vegetatively propagated crops where you don't get the seeds. There you can go for raising the whatever your crop and keeping it as a germplasm collection. So next, the third kind is a shoot tip gene bank. So in this, you will maintain your germplasm as a slow growth cultures. So shoot tip or the nodal segments you will maintain. So it has to be constantly subcultured and maintained. It is like a hectic kind of thing. Okay, shoot tip gene bank. So next coming to cell and the organ gene bank. So here comes the cryopreservation. What is the temperature for cryopreservation?
Minus 196.
Minus 196 degrees.
Liquid nitrogen.
Yeah, in liquid nitrogen we will be storing. So embryogenic cell cultures or the shoot tip, somatic or the zygotic embryos will be frozen and stored. This is the cell or the organ gene bank. So DNA gene bank is your DNA segments, which is maintained as a cosmid clones or the phage lysates, or you can also store it as a pure DNA. These are the different kinds of ex situ conservation measures we can take for the germplasm conservation. So when you have a germplasm with you, what are the activities you can do? So entirely in germplasm collection, we have a set of activities. So from where it starts? As the name says, collection. So you have to collect the germplasm. So collecting of the germplasm for your germplasm collection, storage or conservation, it is a germplasm collection. So exploration. What is exploration?
Ma'am, we will explore the source of collection.
It is the trips for the germplasm collections. You will go somewhere and bring the germplasm materials. All those trips are called as a exploration. So after the exploration, what you will do? You will conserve the material. If it is, you feel that it is not safe or it is in the verge of extinction. So you will conserve that material either by in situ in its original place or outside of its place, ex situ conservation. So meanwhile, you will also go for the evaluation of those germplasm for a set of traits. So what is that? Screening of the germplasm. So variation for this polygenic characters, it is important. While screening the germplasm, if you want to assess the variation for your polygenic characters in your material, you can use these three methods. You just remember the methods, names. So this will be coming in the upcoming classes, okay? There we will deal about it. You just have to remember the names. So simple measure of dispersion we can use to assess the polygenic characters, or you can use the metroglyph analysis. It is given by Anderson, or D-square statistics, Mahalanobis. Okay, these are the three methods which can be used for assessing the polygenic characters. So while screening your germplasm, you will come up with a descriptor. What is a descriptor? It is a set of characters. Either it could be its height, leaf type, or it can be its leaf color, flower color. It is like a set of characters you considered as important. Whenever you get some new accession, you will use those descriptors to define your germplasm. You will keep that descriptors and you will see to which material this accession will belong. Then you will classify them. That is a descriptor. And then you will go for documentation of your whatever material you have collected. So under the documentation, what you have to remember, if it is a code given IC, that is an indigenous collection. This material is coming from the indigenous collection. If it is code EC, it is coming from the exotic collections. If it is high W, it is an indigenous wild collection. So then you will, whenever some person come or a researcher comes and asks for the material from the germplasm, you will distribute it so that they can utilize and give us a new varieties or whatever. So that is a after distribution, it is a utilization phase. So one of the example through the germplasm utilization which has come is the Sudha, which is a coriander variety. It is a collection from the Andhra Pradesh, which is directly released as a variety in 2006. So what are the organizations which are associated with its collection?
NPPGR.
Yeah, all the NPPGR is the one which is associated with the collection. At the global level, that is with India. So at the global level, it is IBPGR. Earlier it was IBPGR. What is IBPGR?
International Bureau of Plant
International
Genetics.
Bureau.
It is International Bureau or International Board?
International board.
Yeah, it is International Board for Plant Genetic Resources. So earlier in 1974, it was named as IBPGR. Then it was changed, the name was changed to IPGRI. What is IPGRI?
International Plant Genetics Resources.
Yeah, very good. It is located in Rome, Italy. Now it is known by the name Biodiversity International. In 2006, it was renamed. Okay. So in the India, collection is majorly concentrated with the NBPGR New Delhi, all agriculture and horticulture crops, and Forest Research Institute, Dehradun. It is for the forest species, and Botanical Survey of India, remaining all other species. Okay. These are the organizations which are associated with your germplasm collection. So next, one of the most important topic, we have the centers of origin. Centers of origin. Who is the first person who is associated with identifying the centers of origin?
Ravilo.
Okay. Anybody have any different answer?
Alfonso Di Candelo.
Heard of this name? Yeah.
Yes, ma'am.
Alfonso Di Candelo. He is the first person who attempted to locate the origin of the crop plants. He has also written a book. What is the book name? Anybody know? It is Origin of the Cultivated Species. Sorry, cultivated plants. Origin of the cultivated plants. It is a book written by him. After him only, Ravilo will come. N.I. Ravilo. So what N.I. Ravilo has given? It is the centers of origin. Centers of origin. What is the center of origin? It is a place where our
more density.
Sorry, I didn't hear you.
Place which having more diversity of plasms.
Yeah, it is a place from where the crop plants have been directly originated. That is a center of origin. So what kind of traits you can see in the centers of origin? It is majorly concentrated with the dominant traits. Okay. And you will see maximum diversity in this areas. So then where do you see the recessive alleles? If we take this as a center of origin, on the periphery, you can see your recessive genes. So in the center, it is majorly dominant genes. So how this recessive genes might have come? Through inbreeding or through the genetic drift. What is genetic drift?
Random change
change in gene population.
I need a complete sentence for that.
Random change in gene population by chance.
By chance. Very good. So that is a genetic drift. Either due to the inbreeding or the genetic drift, it could be recessive genes could have arisen. So Vavilo has given this centers as a primary center and secondary center. What are primary center and secondary center? So a
Primary
place. Yeah, you can tell.
origin as well as maximum number of crop species, and in secondary only maximum number of crop species are there.
Pardon? Can you say again?
Ma'am, in primary the origin plus maximum diversity of that species, and in secondary only maximum number of crop species.
Okay, very good. So in primary, you will see it is a place where that crop is actually originated and showing the maximum diversity. Okay. In the second, you will see only the diversity through the spread of the crop from the primary center. From the primary center, through the distribution of the crop, you will see the maximum diversity here. So one more important law was given by Vavilo while he was analyzing all the centers of origin. What was that law? Anybody know this?
Law of homologous series.
Yeah, it is also known as homologous series, also known as
Parallel variation.
Yeah, it is a law of parallel variations. So what is law of homologous series of variation?
Character found in one species also present in related species.
Yeah, very good. It is a characters which are found in one species is also found in another species. So how many centers Vavilo has identified?
At first 8, then 3.
8. 8 main centers of origin Vavilo has identified. So next comes one more scientist, Harlan. What Harlan is associated with?
Microcenter.
Come again?
Microcenter.
Microcenter.
Okay, good. Microcenter. He is also given some other thing. Anybody remember about the center and non-center? So while studying the pattern of variations, he has observed that some of the crops does not follow this Vavilo variation pattern. That is, he has said that in a primary center, crop has originated. In the secondary center, you will see the maximum diversity. But for some crops, origin is also same, and the place of origin is also same, and the place of diversity is also same. He did not observe the pattern. For some species, it was two different places. For that, he has called center and non-center of origin of agriculture. So it is a main reason. He has given this two centers, center and the non-center. He has just said that center is a place of origin of agriculture or the crops originated. Secondary center is the one where it is dispersed. Whoever the scientist which comes around here, around the center of origin or center of diversity, they all give the same kind of concept for primary and secondary, but their way of defining them is somewhat different. Okay. Center is where you will cultivate your crop. It is in a very limited space, right? It is a narrow area where you will cultivate your crops or whatever. That is where your crop is originated. So it is non-center is like it is a diverse range where you will see the diversity. It is like in a wild condition. This is about the center and the non-center. So we have seen Alfonso de Candolle and in Vavilo, and maximum diversity will be there associated with the wild species and more number of the dominant species. So he has given primary center as it is a geographical region where crop is originated and maximum diversity is there. Secondary is like a high diversity due to the spread of the crop. So law of homologous series of variations, you know. And one more person comes, Zohari. He actually said that center of origin is nothing but it is a center of diversity. They are not the different things. When you hear about center of diversity and center of origin, they are nothing but they are same. He has said that. Whatever the characters you see in center of origin, you will also see in the center of diversity. It is a center of origin is a like a hypothetical place. It is actually a center of diversity which actually exists in the reality. So we have seen this center of diversity. Again, the same comes with the diversity. Primary center of diversity is a place where you have the origin of a species. Secondary center is where we have the diversity. So again, Harlan has given this. Centers and non-centers, we have seen. Center is a narrow area where is crop originated. Non-center is a broader area where it was domesticated. Okay, for the 8 main centers, you can remember the examples wherever it is I have highlighted. You can also remember some other examples which you feel which is important for you guys. It is like unpredictable. They can ask any crop or any place for origin. So you can remember whatever you want. I have highlighted some of the major places or major crops they can ask. Okay, you can go through this. So along with this origin, we have few other scientists. We'll see them. So Zivan and Dewitt. So instead of 8, they told that 12 centers of diversity is there. You don't have to bother about remembering all those places, like 12 places. So you just have to remember the scientist name and how many places they have given. So Zukowski has also given the 12 mega centers are there. So you already told that micro center. Who is the scientist?
Harlan.
Yeah, Harlan. So what is a micro center? You have this is the center, one center you will consider. So within this center, you will have a place where you will get a maximum diversity as compared to this whole place. That center, we call it as a micro center. As the name says, within a center, you have a small area where you get to see the much greater diversity. That we call it as micro center. So hotspot of biodiversity, this you have to remember. So Eastern Himalaya and the Western Ghats are considered as a hotspots of the biodiversity. So what do you mean by core collection? Core collection is given by Frankel and Brown. So what is core collection? So you have a germplasm collection with you. So while dealing with the germplasm, you don't have to consider the whole material every time. So for that, you will create some representative material which contain all the variability which is present in your base collection. That you will name it as a core collection, where if you see this core collection, it is just a representative of this base collection. This we call it as a core collection. Okay, it together represents all the variability. So what is pre-breeding? So anybody can simply define what is pre-breeding? Don't see the slide.
Ma'am, when we introduce a variety from some other places, we first try different method before releasing it as a new variety. So all the processes before releasing that as a new variety are included in pre-breeding.
Okay, very good. You can also say as, so you have a germplasm with you. So from the germplasm, you have many of the materials and diverse collections you have. You cannot directly use them to cross it with your cultivated crop plants. Okay, you want some disease resistance. It is already a well high yielding one. It has all the desirable traits, but you need just a resistance gene. So you cannot directly use it here, either due to the ploidy difference or it can disturb its whole background so that its unique traits may be gone. For that, what you will do, you will first cross it with the bridging species. which make you to transfer these genes to here and from bridging species to cultivated species. So it is all the activities which are designed to identify the desirable segment from an unadopted material. So from your unadopted material, you will take out only those resistance genes. You will screen here only. So, and then you will transfer it into that cannot be used directly in the breeding program, but you will transfer them to an intermediate set of materials. So that can be directly used for producing the new varieties. Okay, those are the pre-breeding activities. It is also called as a germplasm enhancement. So next we have gene banks for various crops in India. So you have to remember the place and the institute name and the crop with which it is associated. And this is the international or the global gene banks for the different crops. So you have to memorize them. So our class will end here. Anybody have any doubt, you can ask me, or you can write in the chat box. If you don't have any doubts, we can end the session here. Thank you for listening.
Thank you.
I have enjoyed the session. I hope you guys have enjoyed.
Yes, ma'am.
Thank you for bearing me on this new year. Thank you. If you don't have any doubts, we'll end the session now.
Thank you, ma'am.
Thank you.